Good morning, everybody. So this is going to be fairly eclectic talk. So we start top down from phenotypic variation, down through evolutionary mechanisms maintaining it, and down to actual variance and how to try to predict or guess causal and functional implications. Please do interrupt and ask questions. I know this is tradition in this room to interrupt and ask questions, but I'd like to reiterate that. I don't want to talk about something which is unclear or something which is boring, and you'd better fast forward me a few slides. So the major interest of this group at MPG is inheritance of complex traits, and I'm pretty sure that this was covered many times in this during this primer course, but I'll quickly go through the major concepts to explain its relationship with evolutionary concepts and functional annotation of variation. So over 100 years ago, I think the debate started between Mendelists and biometricians. In genetics, Mendel suggested that this is the true genetics, is segregation of traits in pedigrees, and this is how genetics works. And biometricians countered, saying that no, if I go to a real population, in this case fish in the pond, and measure something, then I have very different laws of genetics. I have no clear segregation patterns in the population. I have distribution, in this case of a quantitative trait, and this distribution is usually normal and actually obeys some statistical properties which are very common for many different species, many populations, many traits. And this is the true genetics, and it's incompatible with Mendelian genetics. And the idea was that Mendelian genetics is unlikely to explain evolution and is not useful for agriculture, which was vehicle for genetic research at that time, similar to medicine being vehicle for our research today. So what are the laws? And this is depicted by classic quantitative trait being height. So the distribution is usually normal. We know there is variation in the population. This is why it's making it a trait. But what's important is it's heritable. But it's not heritable in the sense that it segregates in pedigrees. It's heritable in the following sense. This is called Galton plot, one of the biometricians. So if we look at relatives, the trait values are correlated. So if we look at mid-parent height and offspring height, we see that there is a very good correlation between offspring and parents. So parental height is predictive of height of the child. However, this has nothing to do with, again, segregation of height in pedigrees. There are big deviations from this line, and so forth. Right? So that's the classic Galton plot. To explain the genetic nature of these traits and explain the behavior of complex traits, we all, till today, rely on this classic paper. This is Fisher's 1918 paper, and I really urge you to read it. It's long and complicated paper, and this is roughly where we're still today on theory of complex trait genetics. The idea is the following, that if we have a complex trait distributed in the population like this, there are many loci influencing the trait, together with non-genetic effects. And the key observation was that if we assume that every single locus follows laws of Mendelian inheritance, segregates in the population, however, additively, they impact the trait, then what we observe all the statistical properties known to biometricians. We would observe heritability, similarity between relatives. We would observe normal distribution. We would observe regression to mediocrity and other properties which people in agriculture and evolutionary biology knew. So what this paper did, it explained genetics of complex traits in the language of Mendelian genetics for individual loci. It's not necessarily true for quantitative traits. We use usually liability threshold model for dichotomous traits. So the way quantitative geneticists think of dichotomous traits is that there is some unmeasured unknown trait called liability, and if liability exceeds specific threshold, there is phenotype is developed. For example, disease phenotype, and the model is exactly the same. Okay, Now, to explaining genetic variation. At this point, usually in my class, I ask students how many parameters normal distribution have. And normal distributions have two parameters, mean and variance. Since we're not talking today about evolutionary biology or about adaptation, we're not talking about population mean. We're interested in variation. So all information about population variation for a normally distributed trait is collapsed in the variance. Right? So we're only interested in single one parameter, variance of distribution of the trait. And variance can be decomposed at the first approximation. And this is the way Fisher dealt with the data. And it's partly tradition, partly convenience that we still think in the same terms. So overall trait variance can be decomposed as sum of genetic variance and environmental variance. To me, it's the easiest way to think about environmental variance to think about residuals. Everything non-genetic we call environmental, even if it's intrinsic biological noise, has nothing to do with environment. So as a geneticist, I'm interested in this, and this is just residual. My friends and colleagues, Peter Kraft is not here today, I hope. So my friends and colleagues from epidemiology department, they are unhappy. They really want to know what is here. So we just call it everything else. What is the idea of variance decomposition? So if we have, for example, 3 genotypes, I think this slide. Yeah, genotypes are not shown. So for example, we have 3 groups of individuals. We can stratify them by genotypes or by some other variable. And the total variance would be sum of variance of means and variance around the mean. Right? So if I can stratify my population, I have mean in every group, and I have residual variance within each group. And the total variance is simply sum of all 3. Using this trick of variance decomposition, I can take genetic variance and further subdivide it in some convenient parts. Some of them are convenient, and some of them are very inconvenient, but frequently neglected. First is called additive variance. So because I think about this individual genes, I think there is contribution of this variance independently of anything going on. Independently of environment, independently of variation in other loci, independently of anywhere else. There is contribution of dominance because we happen to be diploids. And I don't want to talk about tetraploid organism and how complicated this whole story is. But in diploids, of course, I may not explain all of the variance by simple additive effects of individual loci, by just regressing my trait on allelic dosage at each locus, because some action can be recessive or dominant action. So heterozygotes would not be in the middle of both homozygote forms. Then action of loci may not be independent of each other, and this creates epistatic variance or interaction, VI, interaction term. And some mutations can be picked up on the way, generating new mutations. Right? So these are different contributions to genetic variance. Yes?
Shamil, where would you put in gene by environment interaction?
So G by environment interaction, that's an excellent question. So what I usually do, and at least here, I assume that this is evil concept in the following way, that if you are interested in genetic variance, and you have access to whole population, you sample all of the population from all environments, then environmental variance becomes residual. So it's uncorrelated with genetic variance, and gene by environment interaction is 0. So this is what I assume here. Now, in reality, of course, there are 2 reasons, 2 possibilities it's not 0. One is that your sampling is unequal and non-uniform. You sample phenotypic effects in one genetic background, and then you go to a different environment, and your effects may be different. Also, if you are actually interested in specific contributions of environment, not necessarily even environmental, it may be, I don't know, gene by age interaction, then this term exists, and we usually put this on the previous slide here. Okay, so now what I can do, I can take my trait, and I can identify individual effects just by regressing the trait on genotype. And I assume that this was covered in this course, and most of you are familiar with this. So I just regress my trait on dosage of on allelic dosage. And this way, what I do is I'm estimating the effect of this particular locus on the trait. And doing this, I can compute individual locus effects, and I can estimate additive variance. Right? So this is my model now. That's purely additive model. I assume that my trait, this is classic model, and there are many more complex models, but this is the simplest version of linear model in genetics. This is my trait of individual J, and it determined by additive action of every locus. So loci are indexed by index I. So this is genotype of individual J at locus I, weighted by effect size, plus some error term, random noise, environmental, or other variance components which would go here. And I can compute additive variance if I know allele frequencies at each locus and effect sizes. So this assumption is very important. I assume that all loci are independent. That's first order assumption. So as soon as I start treating them in a non-independent way, this creates interaction variance. As soon as I start treating allelic dosages in the same locus in non-independent way, this creates dominant variance. Right? So I can simply compute that, how much variation I have in the sample. Now, we have two very important notions. We have broad sense heritability and narrow sense heritability. So broad sense heritability is how much of my variance is genetic variance. What proportion of total variation, total phenotypic variation, is due to genetics. And narrow sense is how much of total variance is due to additive variance. Right? So this is how we measure whether trait is genetic or non-genetic. If a lot of variance of the trait is due to genetic factors, it has very high genetic contribution. Is very little, it has very little genetic contribution. And the rest is, of course, environmental in our lingo. Now, couple important considerations here. So if I look at this Galton plot, I can look at regression of phenotype of the child and parental phenotype, and computing covariance, this covariance between mid-parent phenotype and phenotype of the offspring actually is half of additive variance. There is no dominant, and quarter of epistatic variance. So what people usually do in genetic literature, they would say, okay, my heritability, and when you see the word heritability, usually it's narrow sense, not always, but most likely it would be narrow sense. So it's only additive contribution of loci. And I would say, okay, I have this additive contribution of loci as fraction of total variance. I can take this covariance, assume that interactions are unimportant, which is a big caveat, by the way. I assume interactions are unimportant, and I assume that additive variance can be approximated by covariance between mid-parent phenotype and offspring phenotype, and normalized by total variance. Now, for mathematically inclined people, variance of mid-parent phenotype is half of total variance, so this coefficient goes away. Okay, so this is the way we usually think about heritability. We take relatives and pedigrees, we measure covariance between them, and we compute how much of total variation is due to genetics. Now we have genotypic information, and being last session at this course, I'm not going to talk about GWAS. I assume that you had multiple presentations about GWAS, but if we have GWAS, what I can do, I can take my phenotype, regress on every locus, every marker on the genome, okay, and compute additive variance and estimate heritability. So in the ideal world, I just do my regression, estimate all betas, call them beta hats, find all loci significantly and reproducibly associated with a trait, and estimate my additive variance by summing contribution of every locus. So in the ideal world, this should give me estimate of heritability, which I measure from epidemiological data. And this is, unfortunately, at this point in time in 2013, and I'm trying to get used to saying 2013, not 2012, it's much smaller than what we see from epidemiological data. Right? And this is called missing heritability. If we think how much we can explain, even in best cases, we explain 10, 12%. Where this missing heritability is, I hope there are no epigeneticists in the room. Right? So I assume that there are three potential explanations. One is we may have many more common variants of weak effects. We just don't have statistical power nowadays to detect them. We may have rare variants of larger effects. So they may have larger effects, but we cannot detect them because they are rare. The frequency is low, so statistical power is diminished. Or it can be potentially, I'm not claiming that this is the case, but it is feasible that some of that is in this interaction term, that heritability was not measured correctly to start with, because this term is usually neglected in all papers estimating heritability. Right? So it can be interactions, can be rare variants, can be common variants of weak effects. And what happened during the last couple of years, and I think you probably heard about this in this series, is polygenic modeling. And why do we have this polygenic modeling? The idea of multiple loci contribute. We are vastly underpowered to detect this, this loci. And this was brought to human genetics primarily by people with experience either in agricultural genetics or in evolutionary genetics. And the logic comes from multiple experiments based on artificial selection. So people in both agricultural genetics or animal genetics try to do, or plant genetics, try to do selection and see how many loci would contribute to a trait. And this is, there are multiple experiments like this. This is I love. The idea is the following. So there are Drosophila flies, and the trait here is speed of flight. So the setup is the following. There is a banana, and you release Drosophila here. Drosophila flies towards the banana, okay? You take fastest Drosophila who first reach banana, breed them, take second generation, and then introduce fan, and start blowing wind in the opposite direction. Some of them can still make it. You take them, breed them, increase speed of the fan, and increase the wind, and repeat. So the idea was that if there are very few loci which segregate in Drosophila population and determine speed of flight, you would very quickly reach saturation. So if there are three loci, you fix one locus, second locus, third locus, and you exhaust all additive variants in Drosophila population. You cannot select any further. So the reality is, and I look at the original paper, at this review, numbers in the text versus number in the plot, this is how it works. You start with Drosophila, which fly with average speed 2 centimeters per second. 2 centimeters per second is maybe Drosophila, and this regular Drosophila you see. And you do the experiment in multiple line, and you end up with almost 2 meters per second. I can't imagine this flies. Right? Yes? I don't know. So I don't know if Olympic Committee was involved or anything like that. But what is amazing that until you give up after 100 generations, you do not exhaust anything. You keep improving Drosophila. So if I don't know how many of you saw a paper on selection in chicken, where hundreds of years are behind us with agricultural selection in chickens, or even apples. So a lot of crops now are still not genetically engineering. We just go and use standard artificial selection. So what it means, it's very hard to exhaust additive variants, most likely because multiple alleles are segregating. They are individually rare or common and with small effects. Or maybe there are new mutations coming in during the course of the experiment, but in this case, the target is very large. Yes?
The assumption that everything is linear is also, as you said, a huge caveat. Once you have nonlinear effects, I mean, your space is nonlinear. You could have negative effects coming in from the interactions. You'd never, the chance that you find the maximum is
Uh,
small.
if there are two different questions here. So one question is, even in an epistatic scenario, if I only have 3 loci with effects on the trait, I'm going to exhaust the system, right? The other
If you have 10, then that wouldn't.
if I have 10, all the epistatic variance makes it much more complicated. Now the question is how much nonlinearity you would need. One of the situations is the following. Maybe if it's going too complicated, we can talk offline about this. So if you think about Breeder's equation, so response to selection, usually in an epistatic scenario it goes off much more rapidly because slope is heritability, is additive heritability. Because if you think about relationship between parents and kids, and you think that all interactions pairwise, I'm not going to triple interactions, then you have this half of variance in covariance because you inherit half of the genome from one parent. But you have only quarter in interactions because you need pairs. The question is how many pairs of loci you inherit. And then the fall off is 1 over R for additive effects and 1 over R squared for pairwise interactions and so forth. Right? So saturation is in many epistatic model actually happens much faster. Right? The other, although there are slightly nonlinear and hugely epistatic model which still have this behavior and are very difficult to discriminate. Second issue is that you can kill all additive variance in epistatic model if you have negative epistasis. Right? So if I have this allele affects phenotypes in this direction, in this background, this also affects on a different background, but if they are together, they actually act down. So in this case, you can have zero additive variance, and all your variance is in epistasis. Now, do we really believe in pervasive sign epistasis? I don't know. Most people would think that it probably doesn't exist, at least in for most traits people analyzed. The bottom line is you can constrain some epistatic models with some experiments by looking at pedigrees, by looking at what you know, and when you know genotypes, looking at pairwise interactions, and by looking at response to artificial selection. However, where I agree with you is that still space of epistatic models which we can constrain, and we have no idea what happens in this space. That's still true, unfortunately. Okay, so this brings us to the main question, I think, in the field. So when we think about architecture of the traits, many loci, epistasis, non-epistatic, and so forth. So this brings us to the question how this variation is being maintained in the population. And stepping a little bit out of human genetics, from evolutionary biology standpoint, there is one main question which was brought by genetics of complex traits, human and animal genetics, to evolutionary biology, is how do we maintain this variation, right? So I have this normal distribution, and I know that natural selection may operate on this normal distribution, and there are 3 ways to operate. It can be directional selection, can shift mean, and I convinced you that we're not interested in mean. It can be disruptive, can increase variance, and that's important for models of sympatric speciation, and we're not talking about those as well. However, the main mode of selection we observe is stabilizing. It shrinks the variance. So selection, because we don't know whether it's direct or pleiotropic, so we don't really know whether this is the ideal trait. I like to think it is because I found that I'm anthropometrically average male. So I like this model of direct stabilizing selection. So if you select towards some ideal phenotype, you should kill all genetic variants, and at the end, everybody should have the same trait. Right? So the question is, why do we maintain stable variation in populations? And there are 2 major answers to that. One is that there are different types of selection which maintain the variation, and another one is that mutation is such a profound force, bringing in new variants to the population all the time, and selection just cannot cope. Right? So selection is not efficient enough to remove all genetic variation from the population, and this results in stable phenotypic variation. So now I have to joke that I'm from Department of Medicine, so I have to talk about relevance to human disease. So the question is, why does a common genetic disease exist? Right? So we know that rare Mendelian diseases are usually rare because natural selection prevents them from becoming common in the population. Why do we have something which has clear heritable component so common in the population? And there are two potential answers. One is that those alleles which are medically detrimental are not deleterious from evolutionary viewpoint, and you can think about diseases of late onset, and I don't think we really believe in this model, and I don't have time to go into details here, but one idea is that trait is being developed after reproductive age, and selection overlooks it. So I think at least I more believe in the model of small pleiotropic effects early in life, which does make this complete explanation. You can think about changing environment and lifestyle. So our ancestors many years ago, ancestral population in Africa was in different environment compared to modern US population, for example, and natural selection is acting in the opposite direction. Or you can think in terms of compensatory effects being balancing selection, so heterozygotes have advantages over both homozygote forms, frequency-dependent selection, or something like various trade-offs, which as geneticists we call antagonistic pleiotropy. The other possibility is that we, as I said, we have mutation-selection balance, so we have multiple genes which would influence the trait if mutated. Selection is relatively weak, so it cannot eliminate all the variation immediately. Mutation rate is relatively high, and we know, and I'll go over this, how many mutations happen per human. And there are many studies looking at rare variants in complex traits. So one possibility is that this is simple mutation selection balance, meaning that common variants would have a weak effect. This doesn't suggest that common variants may not explain all of heritability. Right? This just suggests that if they do, the effects are weak. And those variants of lower frequencies may have larger effect. Okay, so this brings us to very basic forces which shape human genetic variation. And Chris, does it make sense to go through mutation and selection and drift, just to remind people what it is? I hope I will have some time to go through actually the meat of my talk on annotation and variance. So we have one force is incoming mutation, and we have to estimate mutation rate to see if continuous influx of mutation can actually maintain stable variation. Natural selection I mentioned, and that's a very important force. And this too act in conjunction with drift. So if selection is survival of the fittest, drift is survival of the luckiest. So this is random sampling in populations. And it is frequently important to take into account population structure and demography of the human population to explain how alleles together act on traits. So I'm trying to speed up and talk about mutations real quick. So we have estimates of mutation rates now, and all of them roughly come to about 100 nucleotide changes per individual human genome. It's a rough order of magnitude. And some other events, including repeat contractions, and insertions, deletions, and some CNV rates. And I'm not the best person in the room, I guess, to estimate CNV rate. So how do we know how many mutations happen? First estimate came from Nachman and Crowell paper from comparison pseudogenes between humans and chimps. The second estimate was published by Alex Kondrashov by following pedigrees of not even pedigrees, but mostly looking at de novo mutations with dominant autosomal diseases. It's not a lot of genes, but was a sufficient sample size. And I was very happy because errors on these estimates were pretty large, and they look very similar. Now, next generation sequencing studies provide somewhat lower estimates, a little bit lower estimates, in the same order of magnitude. My personal current belief is that main difference here is due to differences between coding and non-coding regions. and sequence context, because mutation rate has regional variation and has context dependency because of multiple mechanisms of DNA damage, DNA repair efficiency, and replication fidelity. And I'm not going to talk about that, but especially context-dependent mechanisms can be order of magnitude, and they result in differences between many functional and non-functional regions of the genome. Genetic drift is another force. So we introduce mutations to population, and then we sample them, and usually they are rare, so they would either increase in frequency or decrease in frequency. And these are multiple simulations. We start with a variant at frequency 50% and go through small population of 100 diploids, and our frequencies fluctuate a lot, and many alleles are lost and some alleles are fixed. And what is key here is that if we run the same simulation with the largest population size, fluctuations become much smaller. Right? So fate of allele frequencies is determined by size of the human population, and the key element here is that efficiency of selection is much greater in larger population compared to small population, because if selection acts on the background of dramatic changes in frequency and a lot of noise, it's very hard for selection to overpower this sampling. So sampling, this random drift and sampling of allele frequencies would be larger contribution to frequency change than selection. And here, selection actually has a chance to overpower this genetic drift phenomenon. Demographic history also shapes frequencies, amount of variation in frequency spectrum, especially in humans. This is a feed from recent Tennyson et al. Paper on exome sequencing project. So we think that there was dramatic growth in population both in Africa and outside Africa. So this study had African American population and European population. However, there was a bottleneck when ancestors of current European population left Africa, and this had dramatic signature on site frequency spectrum. So if we look at how many sites have certain allele frequencies, these curves are different between Africans and Europeans, and we think that they reflect primarily difference in demographic history. Right? So that's another important consideration when we think about maintenance of complex traits, and of course selection is the key factor. And I'll be talking about selection throughout the rest of my talk because selection is important in two different ways. First, it affects allelic architecture of the trait. Primarily this question whether variants are weak effect, strong effect, whether they are common or rare. And this is primarily determined by selection. Also, if we're thinking where functional variation in our genome is, we look at the variation underlying the trait and we try to find causal functional variation. We can use signatures of selection because if we're thinking about fitness effects, alleles can be deleterious, neutral, or beneficial. We know that there's no reason for non-functional allele being either deleterious or beneficial, simply because they have no role in molecular function and potentially phenotype. We don't assume that all variants under selection are responsible for our trait, but at least we can use natural selection as filter to guide us towards functional variation. And here is the slide about importance of natural selection in humans in protein coding regions. So we collected about 450 exomes, was last year, and plotted the following dependency. If I keep sequencing individuals, so this is number of samples sequenced versus number of SNPs I observe, right? So how quickly this number of SNPs would grow. And standard theoretical model of constant population, no selection, predicts the following curve. So if I start here, I always normalize to heterozygosity of one individual. I start with one individual, I see certain number of SNPs, and number of SNPs goes up with additional individuals I sequence. Right, so this is called Waterston sampling formula. In synonymous sites, I see my slope is much greater, meaning that I pick up more and more rare variants on the way. And we think that most of this is due to population growth. Selection of synonymous sites, biased gene conversion, maybe other forces in play. Probably the main effect is population growth, and this crossing is signature of out of Africa bottleneck. These are data on Europeans. Now, if we look at missense changes and nonsense changes, the picture is completely different. Right? I keep finding new nonsense changes and I keep finding new missense changes more and more I sequence. And if I stratify missense changes into computationally predicted damaging, benign, or possibly damaging, I see that this tracks with slope of this curve. Right? So this effect is pretty dramatic, and as we see in human data, at least in protein coding region. Okay, just one sentence. Maybe I'll skip that. So there was the idea to explain that there are two ways to look at the data: one to track allele frequencies with diffusion models, and another one to track genealogies with coalescence. So I really want to talk about annotating genome, and I still have 15 minutes to go. So we look at the traits. We think our problem is that many variants have weak effects, and many variants have small allele frequencies. In Mendelian cases, we have very few statistical samples to deal with. So how can we say that the variant is actually functional and causally implicated in phenotype of interest? I can look at segregation data, so at data within pedigrees, and see if there is a support for this variant segregating within pedigree. And that's actually a pretty good way to deal with it, but it only works for very highly penetrant alleles. It's not fully conclusive. In some cases because of LD with potentially undetected variant. And also, we may just not have large pedigrees. Many people don't for many variants. There are experimental functional data, which is a great way to go about it. The problem is in vitro assays don't exist for many proteins, and you would have to develop one yourself. Frequency in controls is informative in one direction. So if I believe that my variant is involved in, is highly penetrant, involved in very rare Mendelian disease, but it's in 50% of randomly chosen controls, that's probably false. The opposite argument is not always true, because the variant can be very rare, be observed only in this family, and still be neutral. Animal model rescue experiments is another way, also fairly involved, also convincing. So we stay in here. And I think the reason we survive in the business, not because our methods are so good, but just because the problem is so hard. And obviously, you can use computational methods very inexpensively and quickly. First step we do, we look at the variation coming out of sequencing studies, and we map them on genomic annotation. And I should emphasize this point. I don't have many slides on this. This is a technical point, but very important. Watch for multiple transcript and conflicting annotations. So many sequencing data sets, and we compare data sets analyzed by different analysts, and we get completely different variant sets. Not completely different, but definitely not identical. And then we look at functional data. And of course, the first category which you can predict with simple annotation is nonsense variants, so variants leading to truncation. And it's great because most of them are complete loss of function. However, be aware that they are greatly enriched in sequencing artifacts compared to the rest of variation. And also, some of them are located at the end of the gene. Some may be located in the exon, which is commonly skipped. So you may think it's loss of function, and in reality, it's not. Of course, the best is if they cause nonsense-mediated decay. Of course, frame shift deletions are another class.
So if you occur farther along the gene, they're less likely to be problematic?
Well, if it cuts off two last amino acids, then there is a chance it's not as functional as you think, given that it's nonsense variant. Another class is splicing. And I'm not going through these methods, but there are bioinformatics tools to predict splice changes. They are primarily variants in canonic splice sites, or very close to exon-intron junction, or variants in exonic and intronic splicing enhancers, which are much harder to deal with and are much harder to predict. Our major focus is on missense variants, variants changing amino acids. And we believe that if you look at disease databases, about 20% of those are strongly functional. And a lot of them are moderately deleterious and damaging. So what we try to use evolutionary information coming from multiple sequence alignments and functional and structural information if we have available three-dimensional structure. And I wouldn't go through details of our methods and methods by other groups. There are multiple methods now available. I would go through the logic of those and what are major caveats. So first of all, we use evolutionary information. So we look at individual position. And the idea is that if I stay here, variation in this position is informative about which amino acid would actually fit. Right? So I can build phylogenetic tree because the data are not independent. And I have two problems, purely method development problem. First, I have very few sequences. I want to estimate 20 probabilities for 20 amino acids, and I usually have very few sequences in hands, and they are not independent. So I have to take into account phylogeny, and I have to be Bayesian. I have to do some statistical estimation with very sparse data. So this is a set of main assumptions for the analysis, which many people call conservation. So first, we assume a constant fitness landscape. That's a huge assumption. If I have a couple of minutes at the end, I'll talk about this. So we assume that in this tree, amino acid which is good for fish is also good for human, and amino acid which is bad for human is bad for fish. Right? So first, I don't live in the water, and I have a lot of environmental differences, but also there are many other changes in fish which would interact with my change, and this is not taken into account. However, if we make this assumption, we can estimate two things. We can estimate rate of evolution, and we can estimate whether mutation fits pattern of amino acid changes. So this is basic mathematical model in the field. It's called continuous Markov model. Many methods are non-parametric. They don't use it formally, but the idea is that there is amino acid. We'll look at single site. It says it glycine. It has this codon. At some point in time, random point in time, it changes. It stays in glycine. It's an anonymous change. And then it's an anonymous change again, and then it changes to alanine, another random point in time, and so forth. Right? So we have jumps between states, and they happen in random point in time. And if you think about the tree, you run the stochastic process, and you split it in two along the tree. There is a very important property of these models is that they converge. If you wait very long, you have stationary probabilities to observe certain amino acids. And what we try to do, we try to estimate those from data. Not very well, and again, there is a huge assumption that these probabilities don't change along the tree. Second consideration is, of course, biochemical. We can think about protein structure. An observation made by John Malt a few years ago is that most pathogenic mutations are important for stability rather than function. So you would think that you predict change in stability, you predict pathogenic effect. There are a couple of problems here. One is change in stability of folding is very difficult to estimate. And also, cell, for some reason, is not chemical reactor. It may not be very informative. What we use heuristic structural parameters to fit into our predictor, and structural parameters being fed into the predictor together with multiple parameters from multiple sequence alignments, primarily evolutionary rate, estimated distribution of amino acid, distance from human position, and so forth. And again, there are other methods like SNAP, MAP, SIFT, mutation taster, and so forth. Now, where are we going? How well is it working? Depending on the data set and who does the test, it can be 80% accuracy, 75% accuracy, reasonable, not dramatically high. And some high confidence predictions, you can probably ramp it to 90. And in some way, predictions would be better if we have more sequences and more structures. However, there are a couple fundamental problems which I'll try to go through quickly. So first problem is that we have weakly deleterious variation. That may be important for complex traits, but if I look at Mendelian family, and I have a variant in the gene which is under very weak selection, then I see complete conservation. Because this is classic plot from any population genetics textbook. So this is probability of fixation in the lineage as function of selection coefficient. So if selection coefficient in the human population, in the human lineage, is stronger than 10 minus 3, 10 minus 4, means that individuals with this variant per 10,000 progeny, I have 9,999 if I have this mutation. This is nothing of interest to a medical doctor, but this selection is strong enough to keep the variant completely conserved. Right? So I may have variant completely conserved, the selection force is very weak. And this dependence is exponential, and I can add more species in the tree. This is linear effect, so I may not beat it. The second problem, of course, is assumption of independence. So this is one example from a paper we published also a number of years ago. This is human hemoglobin. And if we believe to the database, change of valine in position 20 is pathogenic. And I look at the multiple sequence alignment, I find hemoglobin of a horse, where I see this glutamic acid in position 20. And that's probably not a mistake in the annotation, because if I look at the structure, there is very nice interaction with glycine, potentially van der Waals interaction. And here, there is electrostatic interaction with histidine. So placing glutamic acid in this background would not really work. Multiple examples like that, not necessarily in the same position. I have a possibility of changing position of a salt bridge to another location and still have stabilizing interaction with amino acid change, which would be damaging at a different genetic background. So if we look at phylogeny of vertebrates, now we have 46 genomes, and we look at neutral variants, we see that only about 26% of those don't have potential human polymorphic variant in any of the 46 vertebrates. But the problem is, if I look at pathogenic variants, I still have 8% of positions where variant, well annotated to be disease variant in humans, and some of them may be mistakes, but we believe not all, is seen in one of the vertebrates. And these are probably cis effects. So in collaboration with Nico Katsanis, we found cases where he, morpholinos, knocks down fish gene, ortholog of human protein in zebrafish, cis phenotype, rescues this phenotype with human mRNA, introduces mutation, pathogenic mutation in humans, fails to rescue its functional mutation. However, this mutation in humans is actually wild-type variant in zebrafish. So he puts zebrafish amino acid back, and there is no rescue. So what we think is happening is interaction with other position along the sequence. And we try really hard. We're trying to develop models to take this into account, but it's actually very challenging. So I guess I'm almost out of time. I wanted only to mention, I'll probably skip this part, that there are many non-coding variants which are also important. Many of them are likely to be important for complex traits rather than Mendelian diseases. There are multiple annotation available, and evidence from both human genetics and population genetics suggests that there are tons of important nucleotides in the non-coding genome, and we think that their average effect of mutation is much weaker in those areas. Thank you.